All right. Hello, everyone. Today we are going to be talking about DNA replication. You should have already watched the Ed Puzzle by now and the introduction to DNA replication. You've also had a reading assignment from Mr. England a few weeks ago. Normally around this time, I would also have an interactive activity or game for you guys to do. Unfortunately, all of them no longer work because Flash Player is not a thing anymore. So we're going to have to jump straight ahead to drawing our DNA replication model. So earlier on in the class, you of course did your nucleotides. So what we will be doing will not be as detailed as that. That was just a small section. I wanted to see what you guys knew about the anatomy, quote unquote, of the DNA strand. Now we're going to be looking at how new DNA is formed. For your DNA replication model, you do not need to have it on separate pages. You do not need to have it very detailed, not like this picture here. All of these pictures have been cobbled together from various websites, depending on what it is I wanted to show you. So for the backbone, you can just draw a straight line. For the bases, you can draw a straight line. You can draw boxes. It doesn't need to be anything crazy. You don't need to have it in its complementary base pairings or anything like that. This is just for you to visualize what the DNA replication model looks like. So obviously, when we're beginning our DNA replication, our DNA is in its helix form where it's twisted around. Here in this picture, we have our ladder. And as you can see, something to pay attention to is that we have 3', 5', 5', 3', and DNA, when we're doing DNA replication, runs from 5' to 3' direction. Okay, and we'll explain later why that's important, that the other strand is the opposite direction. So to begin, we have to first unwind and quote unquote unzip our DNA strands. So helicase will actually flatten out our DNA helix and we'll start to break those hydrogen bonds. The hydrogen bonds have a dashed line to show that they are weak bonds and easily broken. We also can see that our sugar phosphate backbone has straight lines. That means that it's a covalent bond, much stronger and harder to break. When the helicase unzips the DNA, it creates this area called the replication fork. And this is very important because this is where the enzymes begin to work and start replicating our DNA. Now, our bases automatically want to reform those bonds after helicase breaks them. We obviously don't want that to happen, so binding proteins are then put in place so that the replication fork does not close back up on itself. The enzyme primase makes a short segment of RNA called the RNA primer, which are these areas here. These primers act as beacons for the DNA polymerase, saying, this is where you start, dock here. Now, once that happens, polymerase actually docks and starts to add on new nucleotide bases for the second strand. It will move in only one direction. It adds nucleotides from the 5' to the 3' end. There's only one DNA polymerase, which means that on the leading strand, the DNA polymerase moves in one continuous piece. Okay. Imagine that you're peeling an apple and you're doing it very slowly. You're trying to get it all in one piece all around the apple. You just keep turning it slowly and slowly and slowly. You get that one long strand at the end. That's essentially what's happening here. It's adding on the nucleotides in one continuous stream, no interruptions. The other side, because there's only that one polymerase, it has multiple spots where the RNA primase has created a beacon. That means that it's going to do a small section from the 5' to 3' end, let go, and then move on to the next section. Hopefully this next slide will help understand that a little bit better. Okay. So as you can see here, on our lagging strand, so our leading strand is the one long continuous piece. On our lagging strand here, we have our green primary RNA primers. So the DNA polymerase will dock here, put in our fragment here, and then jump to the next green one that will pop up here. So let's say if we started with this one. So if we had DNA polymerase here, adding nucleotides, it would jump back to this DNA RNA primer, and then do this, and then jump backwards, and on and on and on. And these fragments are called Okazaki fragments. And there will be another Edpuzzle that really won't have any questions or anything like that, but it will show you exactly how it works with a 3D model of the leading strand and the Okazaki fragments. So once we have finished the strands, again, the leading strand is just one continuous piece. So it waits until the end of the entire DNA strand. But for the lagging strand, all of those RNA primers need to be removed. We don't need those beacons anymore. We need to fill them in with DNA. Because as we all know, RNA is different and it has uracil instead of thymine. So once both strands are completed, the ligase enzyme comes back in and it will remove the RNA primers and it will fill in with DNA nucleotides and seal those bonds together. That way, when it's done filling in those nicks, it will have one continuous strand of DNA. Something that may be familiar to you by now is at some point, the scientists did not know what the model of DNA replication looked like. So they weren't sure if the original two strands replicated, then broke apart so that it was all blue and all pink, meaning that they didn't mix old strands with new strands. There's this semi-conservative DNA replication model where you have our all blue parent DNA here. Okay, it acts as a template strand. It splits apart, and then each one has a new strand added. And that's just simply how it is. So we have two daughter molecules, one that each have a original parent strand and a new strand. If we have time, then I will show you guys the experiment of how they did the semi-conservative DNA replication model and how they figured out that was the one. So today we don't have to worry about it. You just need to know that when the two strands here, they're the parent DNA, replicate, the two daughter molecules, one has the original strand, which is the template, and the other one is the new strand. So that is the end of our DNA replication model. Next, we will be moving into what can happen if we have mistakes. What can happen if we start working on genetically modifying things. So our next class will be on GMOs, and we will be doing probably like a persuasive argument on whether or not your personal opinion, think that GMOs are good or bad. So don't forget to take a picture of your model and upload it to Schoology. Okay.